So my lab studies how we sense our physical stimuli. Over the last 13 years, we've been studying mechanosensation. And if you take a step back and look at all the different sensory systems that we have, olfaction, taste, vision, most of these at the molecular level have been figured out over the last decades. But the sense of touch that relies on gathering information of mechanical, thermal, and chemical nature from the periphery, such as your skin, and translating this into a signal that the brain understands has been not understood very well at the molecular level. So I'm here to tell you that your sense of touch is absolutely amazing. You have an ability to identify indentations that are 1/500th diameter of a hair cell. So that's quite remarkable. In addition, we rely on affective touch for our emotional health. And finally, for the related sense of proprioception that we'll talk about in a little bit, we have this amazing ability to know where our limbs are without looking at it. And so if I can close my eyes and touch my nose, I do this because of proprioception. Or as shown here, one can be blind and play musical instrument, or in this case, Yo-Yo Ma and Emmanuel X are looking at each other but playing very complicated music. My favorite image that illustrates the power of the sense of touch is this one. This is in the Oval Office in the White House, where this little kid has visited President Obama and says, "My friends tell me my hair looks like yours." And Barack immediately bends over and says, "Why don't you touch it?" So in addition to sensing the texture of the hair, which he recognizes, this is actually a very intimate moment that highlights the importance of affective touch. So yes, seeing is believing, but touching is believing as well. So the flip side of this pleasant touch, of course, is pain. And just acute pain is absolutely essential for our survival, but chronic pain, which is pain that persists even when the stimulus that initially causes it is not there, is a major unmet medical need. So all of these senses, touch, proprioception, pain, get initiated by a group of sensory neurons in the peripheral nervous system, just dorsal to the spinal cord, that gather information from the periphery and transmit to the spinal cord. So I've color-coded these different neuronal cell bodies, and one of the key features of the dorsal root ganglia is that they're extremely heterogeneous. So in black is neurons that specialize in sensing temperature and pain. Others in green are the low threshold mechanoreceptors that tell us about touch, and in red are these proprioceptors that I mentioned. And you can follow these neuronal endings all the way into the periphery and see that they have very distinct targets. So for example, in red, proprioceptors are sensing how much muscle is stretched, while many of the fibers that project to the skin are heterogeneous and form these exquisite connections to various cells in the skin. You have this Meissner corpuscle, Merkel cell neurite complexes, Pacinian corpuscle, all different specialized subtypes that give you that sense of touch. In addition, we'll talk about some of these neurons also innervate internal organs that tell you information about your own self and what's going on inside your body. So although electrophysiologically the cellular properties of these neurons have been described for decades, the molecules that help us sense this information was not known. So how do you sense a gentle breeze or a cactus pricking your finger? We knew for decades that our ion channels specialized to detect them. So their job was to sense pressure or temperature, and by the ion flow, tell you information of what's going on. However, their identity remained unknown. So about 25 years ago, David Julius's lab, my lab, and others identified members of TRP or trip ion channels that are temperature sensors. I won't talk about this today. Just because more recently, just 10 years ago, what are the mechanically activated ion channels responsible for sense of touch were not known. This is a little bit of a history of these ion channels. What do we know about them? Actually, it was in the late 70s, the lab of Jim Hutzpath first postulated that this type of ion channel must exist. So how did he know this? He was actually studying hearing. And the idea there was that when they recorded from hair cells, which do the transduction of mechanical force into electrical signal, they measured that the time lag between stimulation and recording was so short that it could not be explained by anything except direct activation of an ion channel. So for example, in the visual system or taste or olfaction, it's a G protein coupled receptor that does the sensing and then communicates that to an ion channel. But that was too slow to account for this. A few years later, Fred Sachs's lab was the first to directly record from these stretch-activated channels, for example, in muscle cells. So we knew that they're in many places in addition to hair cells. But what are the molecules? What's the identity of the molecules that are these mechanically sensitive ion channels? Over the decades, many channels have been identified in various species. The first was in bacteria, also in worms and in flies. However, none of these translated to mammalian physiology. Either the homologs of these ion channels did not exist, or they seemed to play different roles. There were a mammalian potassium channel, but if you know a little bit of neuronal physiology, you know that if we want a channel to activate these touch neurons, we need a cationic signal for it to come in. Potassium channels, when activated, will release potassium and will hyperpolarize instead of activate the channel. So this identity remained unknown till my lab identified these ion channels that we cloned, Piezo 1 and 2 in 2010. So the outline of my talk today is I will quickly go over how we found these ion channels, tell you a summary of the role of Piezo 2, one of the two channels we found, specifically in somatosensation as well as interoception, which is sensation of internal organs. And then I'll tell you that we're also finding some perhaps unexpected roles of its sister molecule, Piezo 1, beyond the nervous system. So Bertrand Coste, who came to the lab to identify these ion channels, took a reductionist approach. So his preparation to record from these ion channels is based on a cellular electrophysiology. So here's a cell that he can put an electrode in to record, and then with the blunt glass probe comes and pushes the cell. Here's a video of this preparation. So if you push the cell like this, and a cell has a mechanically activated channel, it opens and you can record it. So to find this ion channel, he first had to screen a variety of cell types, easy to work with cell lines that we can grow in a dish in a laboratory, to see which cells had this activity. The idea being is that if we clone it from a simple system like this, later on we can look more in vivo settings and find out what they do. So to find the molecule, he used an RNAi screen, and the idea here was that once we knew cell expressed this, we could use bioinformatic tools such as comparative expression of the genomic RNA levels, as well as candidate ion channels have to have two transmembrane domains. I won't go into the detail, but he came up with about 300 candidates that he had to knock down one by one and see which one is responsible for this current. So here's the experiment that Bertrand performed. This is on the x-axis, you're looking at the maximal mechanically activated currents from Neuro2A cells. The top is blue with a very tight error bars, is scrambled, so if you knock down nothing, you see this average activation of these channels. And then each of these circles is a single candidate that he's knocking down. This was his favorite, of course, the first one, and on, to see if the currents are gone. So these are kind of two standard deviations away from the mean. Anything within this probably is not a real hit. But to get this kind of error bars, he had to spend about 3 days on each of these candidates. And so my main job during this time was a motivator because he was getting negative data day after day. But 11 months into the screen, he hit a very good clone. So this was candidate number 72. There was almost nothing known about it at the time. It was called unknown family 38 member A. he quickly renamed it Piezo 1 from Greek piezi, meaning pressure. And as you can see, if you knock this down, this is logarithmic scale, you see very strong downregulation of mechanically activated current. So this showed that Piezo 1 is required for this mechanosensitivity in these cells. This next slide, when he got this result, we got very excited because it shows that it is also sufficient. So here he's transfecting a full-length Piezo 1 cDNA in naive HEC293 cells, which are not normally mechanosensitive. And if he pushes the cell, this is how much you're pushing the cell. The more you push the cell, the larger inward currents that you see. And this is considered whopping nanoamp currents that we're getting from these cells. And this is the quantitation. So this means that Piezo 1 is sufficient to induce mechanosensitivity. Piezo 2 has a sister. It's about 40% identical in its amino acid sequence, and once again you can overexpress it and get this massive mechanically activated currents, very similar, slightly different, what we call inactivation kinetics. You might see that the currents decay a bit faster here. So one of the fascinating things we immediately noticed when we looked at the genomic sequence of the DNA that gives rise to the protein, of course, is that it's huge. So mammalian Piezo 1 is about 2,500 amino acids, and the prediction suggested that they span the membrane, go out and in of the plasma membrane 38 times. So most ion channels do this about 2 to 6 times. So this has more transmembrane domains than any other protein in your genome. On top of that, doing structural studies using cryo-EM electron microscopy, we realized that they actually form trimers. So 3 of these molecules come together to assemble the functional protein. And this is a model of that structure. Each of these individual units, the protomers, are in different colors. And what we call in the C-terminus of the protein is only place where really the 3 protomers meet, and that's where the pore is. The rest, you can see them as ribbons, are these transmembrane domains that extend into the plasma membrane, presumably sensing pressure. One of the very interesting features of this, if you can notice, it has this puckered conformation. So we actually think that they bend the plasma membrane in this opposite orientation of what you would imagine a plasma membrane would look like. And the hypothesis is that any stretch of the membrane will actually flatten this ion channel and let the ions come in. So once we have this protein that we know it's a mechanosensitive ion channel, most of our work has focused on finding what it does in vivo. So we identify the ion channel, and then using both what we call loss of function and gain of function mutations, which means ablation of the channel, or if there's too much activity of the channel, both in mouse model as well as clinical data to identify what they do. We often start with expression. So this is an RNA-scope in situ hybridization of Piezo 2 in these dorsal root ganglia neurons that I introduced to you. And the staining of the Piezo 2 RNA is in brown in a light blue background. And as you can see, many of the DRG neurons robustly express Piezo 2. And this was very exciting to us because this is what we were looking for. What are the ion channels in sense of touch required for that are mechanically activated? We cloned this ion channel in a cell line, and in a way we got lucky that it was expressed in the place that we were most interested in. So we have made loss of function mutations of Piezo 2 in mice. That means we've ablated it. We use different promoters and conditional knockouts to do it. I won't go into details. I love to show this slide because it's the first behavioral experiment that we have done. And this is a very non-sophisticated assay that we call tape on the back. So this is a wild-type animal, and we've put a large white tape on its back, and it immediately senses it. It tries to get rid of it. And here is a Piezo 2 deficient animal, the same assay. It doesn't seem to care at all. See what you can win Nobel Prizes these days for? So this is some quantitation of various somatosensory assays that we've done. So on the left is You touch the paw of an animal with an increasing amount of mechanical force, and in gray you can see after a certain threshold, they start responding a certain percentage of the time. I just wanted to show this to see how profound the effect is. These animals are completely, the knockouts in orange or pink are completely deficient in this. And these are two different assays for somatosensation, the third one being tape on the back that I showed you the video for. How about this proprioception that I introduced you? I want to just say that I think proprioception is perhaps your most important sense, and I'm willing to bet that many of you have not ever heard of this. I already told you this is how you can close your eyes and touch your nose. So all this sense of where we occupy in space is something we take for granted. And the reason I think for this is vision, for example. You can close your eyes and easily imagine a world of a blind person. You can do this with almost all of your different senses. But proprioception, you can't really turn it off. And as I said, we take it for granted. But from these sensory neurons, instead of what we're talking about till now, that sense touch in the skin, from how much each of your muscles are stretched, you're not conscious of this muscle stretch of individual muscles per se, but all of them together give you this image of what space you occupy. And is absolutely essential for simple tasks such as walking, or if you're really talented, can perform tasks like that. So in another animal model, we wanted to test if piezo2 was required for proprioception. And here's piezo2 deficient animal. You can see heavily uncoordinated the way the hind leg moves. And this is a typical sign of lack of proprioception. I'm not going to show too much of this, but we don't just do behavior, which by itself is kind of phenomenological. We do in vivo electrophysiology. In this instance shown in the bottom, our collaborator, Katie Wilkinson's lab, has this technique where she can go into specific muscle fibers, stretch them, and record action potential from individual nerve fibers, the proprioceptors. And as you can see in the wild type, when you stretch these muscles, you see massive increase in firing of these proprioceptors, but in the knockout, we see almost nothing. Actually, 7 out of 8, we saw this response. So we know in the mouse it's required for, piezo2 is required for touch for proprioception. One of the interesting phenomenon was our collaborator, Alex Chesler and colleagues at the NIH, identified individuals who have mutations in their piezo2 that makes them completely non-functional as well. And it's been remarkable parallel between the mouse and the human of how similar the phenotypes are. So these piezo2 deficient individuals don't have discriminatory touch. In addition, they completely lack proprioception. And this video shows one of these individuals being asked to do something very simple. As she's blindfolded, she's asked to walk in a straight line. Many of you will do this very easily, but as you will see in this video, she really struggles to do this. She's harnessed to make sure she doesn't fall. So how about pain? Both mice and humans that are deficient in piezo2 experience normal acute pain, hammer hitting your finger. However, we also wanted to look at tactile allodynia, which means when touch becomes painful. You experience this, for example, after sunburn or injury, but people suffering from neuropathic pain experience this as well, and is a clinically important feature. So I won't show you this, but we have shown that in mice, tactile allodynia is completely wiped out without piezo2. But Alex Chester's lab was actually able to confirm this in the piezo2 deficient individuals. This experiment, and we're very grateful that these folks volunteered for this, is a simple experiment where on your arm, in one imaginary box, you put just the vehicle, in the other one, you apply heat and capsaicin, which causes inflammation. You can look at this with a laser Doppler imaging, both in the control individual and the piezo2 loss of function. You get the red, which inflammation is happening. And then you touch the boxes A and B and ask them which one is painful. So the control individuals, of course, 100% know where the pain is, while the piezo2 deficient individuals have absolutely no idea because they don't feel anything. You tell them you have to guess one or the other. That's how the experiment works. And so they get it right about 50% of the time. I think this is clinically relevant because in my field, lots of work gets done usually in the mice to identify targets for pain, and then when we go to human, for some reason they don't seem to pan out. And so this, we're lucky that these individuals are so eager to help science and they volunteer. So this is not complete, but some form of confirmation that the same mechanism is happening in mice and humans. And we think future medicines, ablating or deleting or inhibiting this ion channel topically might help in some conditions of pathological pain. So I've kind of told you already that Piezo 2 is required for touch, proprioception, some form of pain, and mechanical itch that I won't talk about. Most of these is what we think of exteroception. You're sensing stimuli that are mainly dependent on outside. But over the years, together with Steve Liberless and Alex Chesler, we found that mechanosensation through piezos are also very important for internal senses. So for example, Piezo 2 fibers innervate the lung, and they sense how much the lung is stretched and actually control breathing rate. For those of you in medical school, I'm sure you know of the Herring-Brewer reflex in the Piezo 2 knockout, which is an inflation-dependent apnea. And in the Piezo 2 knockout, for example, this is completely gone. And early on in the life of the Piezo 2 deficient individuals, the main medical problem actually they have is problem with breathing, and this is the #1 reason they go to the hospital. Baroreception, again, folks in medical school know this. There are many ways to control your blood pressure. One of the ways is a neuronal mechanism of doing this, where neurons from the nodose peripheral nervous system innervate aortic arch, for example, sense differences in stretch, and with this control baroreceptor. Interestingly, both Piezo 1 and 2 only together are required for this function. We've also shown that both in mice and in humans, the sensation of bladder filling depends on Piezo 2. Indeed, individuals without Piezo 2 have to go to the restroom on a schedule to avoid accidents. This cartoon helps me remind me and you that although this seems like all solved questions, there's actually many things we still are currently investigating. And a lot of these are based on intuitive thoughts. For example, if you eat a very large meal, you feel very full. I don't know about you, but to me it sounds like a very mechanical sense. And yet most of research on satiety is based on nutrient sensing, proteins, sugars, etc. But how much does the stomach stretch itself regulate how much you eat, when you eat, and how it influences diabetes or obesity is not known. And these molecules are a great tool for trying to understand this novel physiology that many people haven't thought about. One of the directions that we're going is finding now roles of DRG sensory neurons in internal organs where perhaps we were expecting less, or we knew less about what mechanosensation or these DRG neurons are doing in general. So for example, I mentioned already that they innervate the bladder. In addition, these sensory neurons innervate the pancreas, adipose tissue, kidney, and we have almost no idea what information they're listening to and what are they doing with this information. I'll quickly go through one recent study we did of innervation of fat tissues. And so the textbook idea of how fat communicates with the brain is that adipose itself makes hormones such as leptin, which is secreted, goes to the brain listens to this message and controls thermogenesis, or the opposite, adipogenesis, through the sympathetic nervous system output. But we knew that there is some sensory DRG innervation of adipose, and what do they do was not very clear. I'm going to go through this pretty quickly because I don't want the talk to go too long, but we came up with a system to specifically label these DRG neurons that innervate the fat tissues and then image them to start getting an idea of, because it was very anecdotal whether these DRG neurons actually innervate fat. And we found indeed, shown in this white labeling, that DRG peripheral endings come and make very close connections to adipocytes as shown here. But this is actually a really new technique that I'm really excited about, and I wanted to show you a video. So it's a combination of labeling the DRG neurons in the lumbar region, letting them go all the way to the periphery, and then clearing the whole tissue and watching this in real life. So you can see that's the spinal cord. You're focusing on the DRGs, and then you can follow individual fibers going all the way into the fat. And the previous picture of innervating adipocyte comes from this. So you get the long range, the whole system, as well as at a single level connection. It's a very powerful method that we're excited to use for other tissues as well. Okay, but what do these neurons do? We know they now innervate the fat tissue. Again, I won't tell you how we did it, but we came up with a way to very specifically ablate the DRG neurons that only innervate fat. Because if you innervate the DRG neurons going to other internal organs, you can get very indirect effects that you cannot conclude anything. And the main conclusion is not important, but we found actually very strong phenotype. When you ablate this inguinal fat, what we see is that strong enrichment of beiging of adipocytes. As you know, humans don't have brown fat, but they have white fat that can, when induced, turn into a beige phenotype, which is important for thermogenesis. So this tissue level morphogenesis change is associated with upregulation of genes, both involved in thermogenesis and lipogenesis. And interestingly, this is an exact phenotype you would get when you actually activate the sympathetic nervous system that innervate the fat. So I'll just show a cartoon of what we think is happening. So we think sensory neurons are sensing something in the fat, and they're acting as a brake of the sympathetic nervous system. So there is an example of this before. The baroreceptor does exactly that. It senses blood pressure, and if it's too high, it acts to reduce it. So very similar kind of system is at work at knee-jerk type of response to innervation of fat. As many good studies, this raises more question than answers it. What is these neurons sensing in the fat? We don't know this. We actually have preliminary data that Piezo 2 is required for this. So there's something mechanosensation related that is being sensed, but we have no idea exactly how. So we're pursuing this. And the reason I like this is because this is complete new biology. No one knew that this sensory system existed, what they did, and it kind of opens the door to try to understand how this system affects feeding and hunger and obesity. And we're eager to do this for many of the other internal organs that I mentioned. Okay, I know this is a neuroscience symposium, but we're going to take a break from that and just say that mechanosensation is relevant for many tissues. Many tissues experience, sense, and respond to mechanical forces. Endothelial cells of blood vessels experience shear stress, muscles get stretched, red blood cells get indented, and many of these processes had not been studied at the molecular level, and piezos are helping us do this as well. So over the years, I won't talk to you about many of these, but my lab and others have found many different roles of piezo 1, mainly the sister molecule of the one that's involved in sensory system beyond neurobiology. So both in humans and mice, loss of function of piezo 1 causes lymphedema, and we've shown that during development, piezo 1 is required for lymphatic vessel development, mainly valve formation. Happy to talk to you about it later. Others have shown that piezo 1 is absolutely required for regulating bone density. We've collaborated with others to show piezo 1 in skin is involved in keratinocyte migration and wound healing. But in the next few slides, I want to talk to you about its role in red and white blood cells. One of the fascinating things about piezos for us is that there's so much human genetics has made us learn about these ion channels and more about fundamental roles of mechanotransduction that we never thought about. So I didn't know this, but red blood cells squeeze through capillaries that are half their diameter. And people had thought this is simple physics. You get squeezed through capillaries, you get smaller. But what we found through human genetics is that this is an active process, depends on piezo 1, that is involved in regulating red blood cell volume. And the way we found this out was human genetics. Clinicians called us and said, we study this condition called hereditary xerocytosis, which causes red blood cell dehydration, and we found point mutations in Piezo1 associated with this. When we looked at the electrophysiological consequence of these mutations, we found that they are gain of function. What I mean by that is that given the amount of pressure you apply, they give more signal than the wild-type Piezo1. Since then, we've made a mouse model of this, which recapitulates the phenotype. But then we were inspired by the literature on sickle cell, which of course has a deformation of red blood cells, and this causes some protection from malaria. And we tested in mice if this happens. And indeed, Piezo1 gain-of-function mice had some protection against malaria, specifically cerebral malaria. As just quickly shown here, is that Piezo1 gain-of-function animals do not have the penetration of the dye as seen in the wild types. I won't have time to talk about the physiology of how they're protective against malaria. A lot of it we don't understand actually. But this motivated us to ask a question that if it is really true that gain-of-function Piezo1 is protective against malaria, shouldn't there be, just like sickle cell, an allele that's frequently present in places where malaria is endemic, specifically in African population? So we took a bioinformatic approach and actually found an allele that we call E756 delete. It's a deletion of a single amino acid that causes a subtle gain-of-function phenotype and is very strongly expressed in people of African descent. So this slide shows the distribution of this allele. In light blue is the wild-type allele, but in purple we show this E756 delete. As you can see, the purple is almost not present at all in people of European descent and many other areas, but in all areas of Africa we see a very substantial presence, including in African Americans over in the United States here. And the numbers are pretty staggering. So 1 in 3 people of African ancestry carry at least one copy of this allele. So back of the envelope calculation says about 400 million people on this earth should have one copy, and about 40 million are homozygotes. So obviously, with this kind of prevalence, we don't expect this mild mutation to cause any disease, but we thought it might be very useful clinical tool to look at associations. So let me explain how we are looking at this now. So the original diagnosis of Piezo1 gain-of-function is this hereditary xerocytosis, and only about 126, exactly 126 people were diagnosed with this syndrome, and they were all of European descent. And interestingly, this very rare disorder, which is diagnosed as a disease, have, for example, other conditions associated with them. One of them is iron overload, which of course is not a good thing, and in the long run can cause organ damage. So then we asked the question, there's no description of this in people of African descent, but are people who are carriers of this E756 delete have an association with high levels of iron? So Shang Ma in the lab did a very simple experiment. He got blood from local blood bank of self-described healthy individual African Americans. So the first thing he does is just genotypes these individuals for this E756 delete. And just like our bioinformatic approach suggests, in gray and in green, about 1/3 have this allele, and we have a handful, 8 people who were homozygous for this. And then he assays things that are clinically relevant to assess iron levels. So for example, high transferrin saturation levels or high ferritin. And from those individuals is what is the percentage of this allele. As you can see in this pie chart, as you do this, you get a higher representation of people who are carriers for our allele. Interestingly, if you combine the two, which becomes a clinically relevant level of high iron that they should pay attention to this. All 10 individuals from this 333 had at least one allele of E756 delete. So this is rather impressive numbers. E756 delete is 12 times more likely to be present in individuals with high ferritin levels. But we realize, of course, that this is the first study and much larger studies are required. Overall, the studies of genetics is actually telling us something interesting, which again parallels perhaps the sickle cell story, that this consequence of gain-of-function piezo-1 at one way of looking at it is that through its red blood cell deformation protects against malaria. And again, more human studies are required here, but there also might be disadvantages. So I didn't tell you how iron levels are affected, but what we also like is this using both, starting with the human clinical data, which gives us phenomenology, and then we bring it into the mouse and using tissue-specific knockouts, etc., quickly figure out mechanistically how this gain-of-function allele is causing this phenotype. In the iron overload case, we've shown that actually it is because of overactive phagocytosis of macrophages that causes these levels. And again, this is, I think, a great clinical opportunity for us to look at more phenotypes and genotypes from the hereditary xerocytosis, only 126 people, to this large population, and as well as mechanistic studies in mice. So I will end by just telling you that piezos are present not just in vertebrates, but across the phyla. So for example, they're present in fruit flies, in worms, in unicellular organisms, as well as plants. And we had one study from postdoc Ali Reza Musavi, who's an expert in Arabidopsis, to start understanding what piezos do in plants. And plants also experience mechanical force. For example, this is one of many things plants do that are related to mechanosensation. If they experience a harder surface while their roots are penetrating the soil, they have to decide either they want to go through it or go around it. Again, this could have been a passive phenomenon, but actually there's active signaling thought to be involved in this. And interestingly, Ali Reza found that piezo in plants, which is homologous to the mammalian ion channels I've been telling you about, are expressed very highly, especially where you think this is relevant at the root tips. And he actually has shown, this is one picture of plants forced to penetrate a harder surface. So all the wild-type plants can go right through this gel. You can't see it because of the way it's focused, but if you do not have piezo, they cannot penetrate and go around it. And again, because of the focusing, you can see the roots here in this photograph. So again, this is just a reminder of how amazing evolution is, that the same ion channel that we use to sense touch, plants use to sense their soil environment. I will end there just to say thank you for all my current and alumni over the last 23 years and collaborators across the globe. And this highlights in blue the folks that did the work that I described in this seminar today. Thanks for your attention.
Thank you, Dr. Patapoutian. Now we are going to have a round of questions. We have 15 minutes for that, so as many questions as we can. So we're going to ask everyone to make them concrete and really quick so that everyone has a chance. If you want to make a question, please raise your hand.
Thank you, Professor Ardem. It was very interesting, your presentation. And I'm talking regarding what we all speak as translational medicine. And comes to my mind throughout my practice the cases of two children with what we then called familial dysautonomia, the Riley-Day syndrome. These two children were of different ages, and I followed them for at least 10 years here in Quito, Ecuador. They were of Ecuadorian extraction, Quito and Ibarra, the families. But these two children were very ill, hyperactive, no sensation of touch at all, or temperature, and because of their hyperactivity, they were prone to accidents, burning, destruction of their own tissues, and all this. With the idea of our students here, how will we fit? I know that nowadays you have a new classification of these diseases, the hereditary autonomic neuropathies and sensory neuropathies, and there are many genes involved. How will this fit to improve the condition of these type of diseases that we still see? Thank you.
No, that's a very interesting point that you bring out. I think one of the lessons I learned is that, again, all these individuals that were Piezo 2 deficient were initially misdiagnosed. In the clinic, this lack of coordination is usually thought to be due to defects in motor neurons or muscle dystrophies. These are the common reasons. No one thinks of the sensory component. And it took genomics and sequencing these individuals and saying, there's a big literature of this gene involved in touch, so let's do sensory phenotyping. And when they did that, it was very clear that this is what's going on. So how does this actually help? There's a short term and a long term. In the short term, we actually found out that some of these individuals were given wrong advice. So if you don't have proprioception, the way you would compensate for it is visually, right? And so these individuals, for example, had learned that the way they can walk is to look at their feet when they walk, because otherwise you don't get the information of where the feet were. And therapists were telling them to walk like, control individuals. Don't look down, look up. And that's, of course, the absolute wrong thing to tell them. So just understanding that it's a sensory problem, you can immediately, without drugs, just try to realize that you have to use your visual system to compensate for lack of proprioception. But I think in the long term, what I'm finding very, very beneficial is this collaboration with clinicians and basic researchers. So we have such a good thing now going where the clinicians, keep giving us information about these individuals, their ability to control their bladder. We have projects in kidney function, and then we can bring this back and very easily in the mouse understand, as I said, mechanistic aspects of it. So I think collaboration between the clinic and basic science is going to be absolutely essential to make this go forward.
So, thank you for this very enlightening presentation. My quick question, because I'm picking up from something that is amazing that you mentioned, that even endothelial cells may have Piezo1. So one of the mechanisms of autoregulation in the cerebral blood flow is that as the blood pressure goes up, for example, the vessels constrict. And the way they do it is a very complex way that is transmitted from capillaries and arterioles all the way backwards, to use the surface, the parent arteries. Has anybody looked at that specifically for the brain? Because the autoregulation is very specific for the brain.
People are actively looking at that. There's some amazing new techniques of looking at capillaries in the brain and see how blood flow affects the dilation and constriction. And piezos are expressed in those. There's actually a paper I was just reading. They've done all the prep work. The big next question is how much, if you get rid of piezo, what happens? And that's something that's happening right now. But the myogenic response, all of these seem to depend on piezo1 in endothelial and the smooth muscle cells as well.
Thank you.
Good morning, thank you very much. It was very interesting. I understood that piezo receptors are involved both in affective touch and also in chronic pain. On the other hand, I was thinking about oxytocin, that is a neuropeptide, that it's also involved in physical contact and plays a role on pain modulation, and it's a target for chronic pain. So is there a known mechanism how piezo receptors are related to the limbic system or maybe to the oxytocinergic system in the relation of physical contact and modulation of pain?
Actually, I will go back and tell my lab that you asked this question because I've been asking people to look at oxytocin levels in response to a wild type and piezo2 knockout specifically to start exploring that direction, but no one's done this yet. We should do it.
Thank you very much, Dr. Ardem Patapoutian. Again, you inspired us with this lecture because you brought up the issue of fat innervation, which is just a very, very tantalizing concept. We've looked at fat for many, many years as just a storage material. Then we found that it is a major source of stem cells. And you mentioned about the secretion of certain hormones. Now with this innervation in these DRGs, the question I have is the growth of obesity or the consequence of obesity, can that be modulated with mechanosensation with these piezo ion channels? Or is it still majorly a hormonal or nutritional aspect that is controlling this?
Yes, we're going to start a company that puts a vibrating belt on your fat and you're going to lose weight. It's going to be great. Very interesting. We don't know, but that's why we're such an early stage of this. But that's exactly the whole point, is that there's one wing of communication between fat and brain that was completely ignored, and we kind of know the baseline of what it's required for now. We have a lot of more work to do to understand it at the molecular level, and then start thinking about more translational work.
Dr. Patapoutian, fantastic talk. The question I have is, in the setting of a pathology, like a CNS pathology, which can be induced in animal models and or in humans who are experiencing it, has there been any data showing up or down regulation of the mechanosensitive channels?
Unfortunately, very quick answer, no, but that's not because that's not a relevant direction to look at. I don't think it's been looked at yet. Expression is a great way to look at this because I call piezos professional mechanosensors. They don't seem to be on unless there's a mechanosensory role to be had. Unlike many other channels that we've studied before, they're polymodal, they respond to this and that. These guys, you could see also from the structure, they're evolved to sense membrane tension. And so expression right now has been a very good predictor of function. Wherever we've seen it, there's been function. And this idea of when is it induced or repressed in response to pathological conditions is something we're just starting to look at, and I think there's a wealth of information to be gained by that.
First of all, thank you very much for your talk. It was really interesting. I just want to plug here how genetics can take you to the Nobel Prize, because I'm the lecturer of genetics here. So for my students, that's how you go to the Nobel Prize, genetics. But okay, sorry. Just two short questions. The first of all, when I look at the patients that have piezo deficiency, I mean, I can see the proprioception, but how about the other areas that piezo works in? Like, do they eat more? Do they eat less? What is the consequence of that? And the second question is, I see that piezo 1 and piezo 2 only have a 40% similarity, which makes me think, like, why are they evolving that way? Do you have any evolutionary background? Because if we do have them since we are like plants. What is making them change so much? Because 40% is a lot of change. So thank you.
So the first one, it's a growing story. So I already mentioned they lack discriminatory touch. So if you blindfold them, you touch them, they don't sense anything. I told you about the bladder stretch, they don't sense that. They're actually required in the males for sexual function. That's not unpublished, that's not published yet. And we keep growing this information. So the stomach is a tough one, because one of the things we're finding out is that the language you use with these folks could be very deceiving. So you can't just ask them, do you feel full? Because of course they're going to feel full because nutrient sensing does work. And but for them, it might not have this stretch sensation that we do, but it's very hard to still describe it. So we're trying to figure out how to best approach this. So there's many more questions. Do they sense texture in the mouth? All of these are going to be very useful to look at, and hasn't. With respect to your second question about the homology and why do we have two, one of the big differences between them is inactivation kinetics. I mentioned this very quickly. Piezo 2 is much faster to come back to baseline before Piezo 1. And that might not seem very impressive at first, but we think that it's very important from genetics again, because both in human Piezo 1 and 2, the so-called gain-of-function mutations that I was talking about, the only thing that seems to change is the inactivation kinetics. So the human gain-of-function Piezo 1 closes slower. That slight difference is causing some pretty serious symptoms. In Piezo 2, I didn't even talk about this, causes arthrogryposis, which is a developmental deficiency in joint formation. And so this tells me that the inactivation kinetics has to be very tightly regulated, that you have a small effect and you get disease. So perhaps that's why you need two, because that gives you some more flexibility of how much ions to go through given a stimulus. And for neurons that you have easy amplification, you want to use the quickly inactivating one in other cells where it depends on calcium per se. You might need more signal and uses Piezo 1. It's just a hypothesis, very hard to prove this.
We have time for one last question. Does anyone?
Hi, good morning to all. It's Alejandro Lopez, medical doctor and alumnus from the Universidad San Francisco de Quito. Thank you for your amazing talk. So my question is, in your practice, your team and you, have you ever seen compensation in the other ion channels after the Piezo have been knocked down? I mean, because you know, in medicine, when one thing goes wrong, there is a lot of compensation. And as you demonstrate us, there's like 72 another ion channels. So my question is there any compensation in one of them in order to give back to the normality and equilibrium to the system, to the body?
So in a way, we've been lucky. You're right. Usually there's lots of compensatory forces, especially we were worried about between Piezo 1 and 2 because they do very similar things. So if you knock one out, you would think the other one get upregulated and mask any phenotype that you would see. In most cases, we haven't. So it seems like they're specialized, they do their different roles, and they can't compensate for each other. That's why we see such severe phenotypes. But again, I went through this very quickly. The exception is baroreceptor. So in the nodos, there are neurons that express either Piezo 1 or Piezo 2. And if we knock out one, we don't see any effect on baroreceptor function. Only if we knock out both Piezo 1 and 2, we see baroreceptor function go away. And again, this parallels the human genetics very well because this is one phenotype we haven't seen in the individuals, the humans, without Piezo 1 or Piezo 2. Of course, there's no one on this planet that lacks both. And so, again, the mouse and the human agree, but the main answer to your question is they seem to be doing their own things. There's very little compensation except for the baroreception that we found. Thank you again.